In 1953, Watson and Crick created a model for DNA structure. This structure includes base pairing between two strands of nucleotides that allows for constant width and helix. DNA is also composed of a negatively charged phosphate backbone with a ribose sugar and nitrogenous bases. DNA stands for deoxyribonucleic acid. The Watson and Crick model is based off of beta DNA and is right-handed. This means that the DNA coils to the right. It is also important to note that the DNA strand always runs 5' to 3' and anti-parallel to one another. This is an example of two strands running anti-parallel to one another. Both run 5' to 3' but in opposite directions. DNA is composed of bases that consist of adenine, thymine, guanine, cytosine, and uracil, which is only found in RNA. Bases can be classified into purines and pyrimidines. Purines consist of adenine and guanine, and pyrimidines involve cytosine, thymine, and uracil. The base pairing is accomplished through hydrogen bonding, which is relatively weak. Adenine pairs to thymine with 2 hydrogen bonds, and guanine pairs to cytosine with 3 hydrogen bonds. I am now going to build the molecular components of bases, sugars, and phosphates in DNA, and this key will serve as a guide to the atoms in the structure. This is the base pairing of adenine and thymine with 2 hydrogen bonds. This is the base pairing of guanine and cytosine with 3 hydrogen bonds. Nucleotides are composed of a sugar, phosphate, and base. The ribose sugar connects to the phosphate backbone in the base parent DNA. In the sugar, the first prime carbon attaches to the base. The second prime carbon distinguishes between RNA and DNA. The third prime carbon bonds to the next 5' phosphate, and the 5' carbon is where phosphate is initially added. This is the structure and location of the ribose's carbons. In a nucleotide, the sugar and phosphate are connected by a phosphodiester bond. The 3' will bind to the 5' end of the next nucleotide to create a double helix structure that is negatively charged. This is the structure of a nucleotide running 5' to 3' with an adenine base. DNA also consists of major and minor grooves. Major grooves are larger with more space for proteins, while minor grooves are much smaller. Here is an example. Watson and Crick found that DNA had hydrophobic bases with a hydrophilic backbone. Hydrophobic interactions found within the DNA are the main force in stabilizing DNA, and is maxed due to base stacking. This helps handle the negatively charged backbone, while hydrogen bonds mainly provide specificity. Base stacking orients bases on top of each other to prevent any water from entering the hydrophobic core. The DNA melting curve shows the denaturation of DNA through various stages. The TM is the midpoint where 50% of DNA is denatured, and typically a higher GC content will result in a higher TM. In a high salt concentration, the cations of the salt will shield the negative charges of the phosphate that destabilizes the DNA. This will increase the TM of the DNA since the salt helps bring stability.